Now we move on to Dr. Doreen Goh from Sabah. A brief introduction about Dr. Doreen Goh for those who are not familiar with her. Anybody who works on teak, especially teak tissue culture, would have definitely come across all her papers, which talk very elaborately about tissue culture, the process of standardizing the protocol, the process of commercializing it, and how it has been taken up on a global scale. And now we have so many teak tissue culture plantations around the globe. So a brief introduction about Dr. Doreen Goh. Dr. Goh graduated with a PhD in molecular biology, Department of Entomology at the University of Kentucky, United States in 1988. She went on to do a postdoctoral associateship at the North Carolina State University, specializing in molecular studies of the baculovirus as biocontrol agents for the tobacco industry. Since her return to Sabah in 1995, she started her employment as the principal forest officer in the forestry division of Innoprise Corporation, the commercial arm of the Sabah Foundation Group. Under her direction, the plant biotechnology laboratory that was established as a joint project with CIRAD, a French research and development organization, evolved into a full-fledged commercial subsidiary from 2003, which is continuing its service until date. As the managing director of the YSG Bioscape, Dr. Goh continues to publish scientific papers, offer training to practical students from local universities, and manages the day-to-day marketing, production, and supply of tissue culture clonal teak and other cash crops to both overseas and local markets. I now request Dr. Doreen to make her presentation.
Okay, thank you very much. It's a pleasure for me to be invited to attend this webinar. My sincere thanks goes to the organizing committee of IFGTB and ICFRE, especially to Dr. Yasoda, who has been in close communication with me regarding this webinar. So I'm pleased to be here. Thank you very much. I think with that, I can start my presentation. I can have my first slide. Can you all hear me okay?
Yes,
yeah,
ma'am.
okay.
Okay, good. Okay, I see all the thumb up here. That's good. So it's about 1:30 here, 1:30 p.m. And unfortunately, the Wi-Fi in my office broke down. So I had to rush home to set up everything. And now I'm at home, but there's a grass cutter outside making a lot of noise. So I hope you don't get to hear that. It's my presentation. Okay, good. It's coming on. Okay. So that's good. I hope I can just click and go forward. So I can start, right? Am I okay to start right now?
You can start, but your voice is very feeble.
Oh, okay. So let me see. I think my speaker is on full blast. Can you hear me now? Is that okay? No?
Ma'am, you can go ahead, ma'am.
Okay. Okay. Again, thank you for the invitation to present my work for the more than 25 years of work here at the Sabah Foundation. I'll be talking today on the production, cultivation, and conservation of teak clones. I think it was interesting to listen to Dr. Ma earlier because I'm finding out that what he spoke about, I can generally complement our work with what was presented by Dr. Ma. So hopefully my presentation will help make the way forward where teak plantation or sustainability of teak is concerned. With that, let me see. Where do I go? Okay. So we know earlier from the earlier talk and us in general, we are facing a crisis in the supply of natural teak wood from indigenous countries in Asia. And now a lot of plantation logs are actually coming into the picture. So you see a lot of emphasis now on fast-grown, small diameter planted log coming from Asia, Africa, and Latin America. And as mentioned earlier in the previous talk, we are looking at the current teak forest estimated at 4.35 to about 6.89 million per hectare. I'm going forward. Okay. So in general, we know that plantation in the past have been using planting material from seed sources. And there's, of course, a lot of disadvantages using seeds, such as insufficient quantity, unpredictable and poor germination rate, unknown genetic and quality, leading to a lot of variability in the plantation. And as a result of this, we at YFG Bioscape and jointly with CIRAD, a French R&D organization, decided to embark on propagation of clonal teak. I don't seem to be able to. Are you navigating for me? I'm sorry. Okay. Am I doing it for myself?
Yes.
Can I do it for myself? No.
Yeah, yes, ma'am, you can do it yourself.
I just click it and it will go forward. Is that right?
Yes, yes.
Okay. So we decided to embark on the clonings of teak. And this was a project set up in 1993 on the tissue culture aspect between Sabah Foundation and CIRAD. But then prior to 1993, CIRAD was already helping Sabah Foundation together to come up with a, okay, here you go, I take control. So nursery methods in vegetative propagation of teak was already commenced earlier than 1993. Now, on the research on teak, we wanted to focus specifically on outstanding phenotypic qualities, which means we got to the tree, to the field. We look at teak trees, and based on the phenotype, looking at the vigor, the growth rate, the bole shape, amount of branching, wood quality, and so on and so forth. We then select the mother plant and then bring it into the lab. So to start off here, we have a stock of plant mother trees in one of our research centers in Sabah. Now, the source of these mother plants are from seeds originally from Solomon Island. Out of these trees that were planted in our research plot, we selected 8 clones. And the 8 clones at that time was about, about 13 or 10 to about 13 years, not very old, but then they look pretty good. We knew that the stronger the intensity of selection, the greater the commercial gain would be. So thereafter, as you can see, we were able to, bring back vegetative shoots from standing trees, put them under tissue culture by using nodal segment, and then from the nodal segment, axillary shoot that were produced can be further multiplied. And we managed to come up with an optimal protocol whereby high multiplication rate can be achieved. And I think it was important for us to focus on a very simple, very cost efficient and sustainable method, which will give you to a homogeneous growth and very predictable yield of the planting materials. So for about 2 to 3 years, we focused on the development of this protocol. And in no time, we managed to come up with the ability to mass multiply all these clones. And currently, the capacity in our lab is about 1.5 to 2 million plants a year. But then we only produce according to orders. It depends on, we only produce only when there are orders coming in, confirmed orders. You know, it's all about cost. Of course, tissue culture is never cheap, so we have to be very much cost conscious about you know, the way to go forward with this mass multiplication. Having done that, also, beside the introduction of segments, you know, from vegetative shoot, we also managed to do what we call initiation of meristem culture, and these meristem are quite small, 100 to 150 micrometer in size. But the good thing about teak is, even though they're small, we can still see it under a light microscope. This method of meristem culture helped to remove endogenous microorganisms, and this would lead to healthier plants and also, you know, more vigorous growth. So from this early work on meristem culture, we put them out in the field to test them. And this is a picture of an 18 months meristem issued teak tree established in the field. And this other picture is a 7-year after planting, showing a girth of about 55 centimeter and a height of about 25 meters. So statistically, based on our analysis, we know that meristem culture plant from 3 of the Solomon clones combined together show significantly larger diameters and volume than the same materials produced for microcutting. In other words, the meristem cultured issued trees are more vigorous and they grow faster in growth than, you know, the other tissue culture plant for microcutting. So this was very interesting for us, and this work has been published by Montuis, who is our lab collaborator at that point in time, in various paper. So, you know, being able to do vegetative propagation by cuttings and also meristem culture really help us to move forward in the vegetative propagation of teak. So from the lab, of course, it's important to find out whether the tissue culture plants are going to be successfully survive out in the nursery. And here, as you can see, we we observed that the acclimatization of a plant in the nursery was actually very much adaptable. And we come up, you know, for us, it's necessary to come up with more than 90% survival in order to really say that the technique works. Rooting of tissue culture plant usually takes place 3 weeks after the transfer from the lab into the misting culture. And from the survived plants in the nursery, it was also important to test the plants in the field. And we decided to do that in early days under different site conditions in Sabah. It was important to test this out in different areas in Sabah, so that we know for sure that the plants that are produced vegetatively are true to type. So here we have a comparison of seed-derived and clonal plants in a trial, and this picture was taken at age 7. I put down here the condition just for you to follow, you know, maybe give you some idea of the kind of conditions that are being used when we were testing all this clone here. Here, under Sabah condition, you know, our mean annual rainfall is quite high, averaging at about 2,500 millimeters. And we do not have a distinct dry season as in India or other places where teak grow natively. The temperature is, of course, about 26 to 28, although now with global warming, I think we are seeing more and more of a temperature in the 30s. And of course, rainfall continues to be quite heavy for us, and we are reaching about 3,000 millimeters per year now. The soil is 5.7 to 6.5. It's a broad range because normally out in the natural environment, you know, the pH is never really specific in tropical countries as in Sabah. So Here's another comparison, a trial of the 8 Solomon clones planted in 2 different sites in the same region, in the same area in Sabah. And I just want to bring your attention to the mean annual increment. On site 1, the MAI for height is about 3.2 meter. The DBH is about 3.2 centimeter. And conversely, on site 2 is not that much different, showing that the plants, the clones, despite being in different area, different sites, apparently are still giving almost similar yield. Against the climatic conditions for this area here is not much different from the earlier site. Temperature 2,500 millimeter per year, no distinct dry season. So just make a note of this, that the MAI of the height and the diameter of these Solomon clones, you find out as I progress in my presentation that the growth rate of the clones are going to be quite similar in wherever we plant them. So the informative figures from the first clonal trial established in Sabah. This was published again in some of our papers. Roughly, just if we are looking at a 7-year rotation density of 1,111 trees, which is 3 by 3 meter spacing, you'll be looking at about 331 cubic meter per hectare, or 47.3 cubic meter per hectare per year, which is quite good. And this alone is for clone 3, we call it Solomon Island clone 3. And then there's another clone, also Solomon Island. We come up with an estimation of about 244 cubic meter per hectare, or 48.8 meters cubic meter per hectare per year. And what is interesting is for our 10-year-old teak, when we cut them, we noticed a very high hardwood to sapwood content. And this was an exciting find for us because a lot of questions came into play in the early days when we started this work on whether we are going to get enough hardwood to substantiate the planting of teak in a very wet area like Sabah. But the wood cross section here tells us otherwise. To further our propagation of teak vegetatively and testing them on different sites, advanced research was carried out with CIRAD, and these were all early works in the early years. We did things like destructive sampling, collecting plants from different individuals of the same clones. Of course, once you cut the tree down, you are literally, removing the tree. So it's important that you have the same copies of the same clone, a different copy of the same clone to do this kind of destructive sampling. And we also have what we call cold sampling of standing tree. And using near-infrared spectroscopy, our colleagues in France, they managed to come up with a very reliable and versatile analytical tool which can handle different wood sample and can detect chemically induced wood duration. So this was quite useful, so to speak. And at the same time, besides wood analysis, we were also looking at DNA analysis. And here we managed to do DNA fingerprinting of all the 8 clones under commercial production. DNA analysis, of course, as you all know, will allow better characterization of the selected clones. It can help to determine the origin and the genetic diversities of the clone, help to assess the site and genotype interaction, and then, of course, certify whether the technique that we are applying will be true to type when we plant the trees out in the field. Now, in terms of wood analysis, of course, when you look at wood density, strength, and so on and so forth, we know that wood harvested from plantation will never be as good as those from natural forests. But then, based on the analysis that was done, we know for a fact that it's actually not inferior at all. So the wood analysis for each of these clones was, in fact, compared to a sample taken from a 40-year-old wood sample from Myanmar, and the data that was shown on each of these what we call flyer sheets for each of these clones was quite impressive. So it was not bad at all. So having DNA and wood analysis, we in turn can use the result from this kind of analysis for certification and for marketing purpose. And in the event of buyers, or some unscrupulous people getting hold of our clones without permission from us or propagating them, then we can always fall back on the DNA analysis. So having done all this preliminary work, we started to dispatch or sell plantlet to many countries across 4 continents. Of course, a lot of these people, through our work and from conferences, they got to know about us, and then they approached us and then wanted to try out the clones. And in the early days, it was a difficult decision. We are all very paranoid about losing control of our planting materials. But I must say we have been lucky or fortunate to associate with very professional people. And these people, they respect document, agreements, contract, and so only then we were able to do what we had been doing. So one of these countries is Australia, and as you all know, Australia has very strict quarantine condition, and it was not easy. The first batch of plants that was sent to Australia was a complete disaster. We lost about 20,000 plants because the quarantine people didn't know how to handle the plants, and they opened up the containers, and all the plantlets, being planted, just completely dried out. But gradually, as time progressed, we managed to learn and teach, show them how to handle these fragile tissue culture plants. And here, as I said, some of the people who approached us, one of them being from Brazil, and in the early days. So this was one of the clonal plots established in 2003 in Mato Grosso, Brazil. You can see here on the left side of the screen, clones from YG, and then on the right side is the seed-derived sources of planting material. In Brazil, for many years, they've been planting teak, but then all the planting materials have been from seed sources. They have very good seed source, I must say, but then they decided to look for alternative aside from the seed because of the inbreeding and not much improvement to the yield. So that was the time when they approached us. So the picture shows at 3 years and at 8 years, and you can see the distinct or significant difference between clones and seeds. And this is just a data from 9 years old, showing the difference between the volume of clone and seedling and the DBH. And as you can see, definitely the clones outperform the seedling in all cases. So they did a rough calculation, and then they estimated that clonal forests can easily outperform seedling forests by 50% in total volume and 48% in DBH, both under the same site management condition. And so I think the idea of shorter rotation in this case of about 15 to 18 years came to mind when they look at this kind of data. Again, at the same time, this was established in 2005 in Australia, in North Queensland. They decided to do a comparative trial between YHG clones and Thailand clones. And interestingly, the high DBH volume, of course, all was a lot more outstanding than the Thailand clones. And here I'd like to highlight to you that the precipitation in the area planted was about 3,000 to 4,000 millimeter per year within a 4-month period. And then after that, there will be a kind of a dry season followed by another 3 months of wet season. So the condition here is a lot different from that in Sabah. Here is another data collected from Mossman, Northern Queensland, showing the YFG Solomon Island clone number 3. This was, I think, a plot of 10 trees of the same clone, and it shows a DBH of about 21.8 centimeters, total height of 19.9 meters, and a ball length of 12.6 meters, which is quite good. You know, when you talk about a harvestable ball, you definitely can get it from here. And what is very nice about this clone is you can see the very thin branching, and it's sub-pruning, quite tall. And for sure, you know, even here locally, we are seeing a clear ball of about 6 meters. This once again proved that the Solomon can adapt to the situation over there, you know, regardless of the lower precipitation of about 2,000 meters, and you know, concentrated only in certain period. In Tanzania, we did a collaboration with the KBTC plantation, Kilombero Berry Tea Company. This plot was established in 2007 on flat and deep soil with varying fertility. Now, the mean annual rainfall in this area is about 1,000 to 1,200 millimeters with a marked dry season of 8 months. This is a totally different area. So after 1.5 years, again, clonal plants was observed to outperform seed-derived plants by 27%, and there was definitely less variation among the clonal trees. Age 3.5 years, average height is about 10 to 11 meters, and DBH is 11 to 14 centimeters, giving almost similar result to what we obtained in Sabah. You have to understand that in KBTC or in Tanzania, because of the distinct dry season, a lot of apicomic shoots are formed, and the clones are not spared from this. You can see a lot of knots on the wood, you know, on the ball of the tree. But I think this is where management, silviculture has to play a role in, you know, pruning or removal of the apicomic shoot. And this is again a management decision on how to, you know, handle this kind of issue. Okay. Back up. Okay. And one last slide on the testing it. This is was taken in Mexico on very good site. This is at age 7 years, and it shows a girth of about 31 centimeters. And this plot was established in 2013. Again, very nice growth. So from here, having a little bit of a problem flipping my slides. Okay. So moving on, after the production of teak is concerned, moving on, now I'd like to talk a little bit about cultivation of the clonal teak. Based on all the information derived so far, we know that our clonal teak is able to adapt to different conditions, different regions across the world. So when we talk about cultivation of teak, of course, we have always got to remember, the factors such as site conditions, source of planting materials, the management cost, harvesting, marketing, and this was, I think, highlighted by Dr. Ma in his presentation to a certain extent. The factors for cultivation of teak will vary from place to place. There's no specific standard or one-size-fits-all program available. So a lot of people will come to me and ask me questions like, what should I do? Can I grow your clone? It's hard for me to say yes outright, despite the fact that I know the clones do well. I will still ask them to say, I always advise them, go find out your site condition. What is the pH? Do you have enough soil depth? What is your precipitation, your drainage? These are all important factors you have to think about. It's not just about getting good planting materials and just planting them. You still have to know all the existing conditions. So with that in mind, in this case here, because of we are able to produce quality planting material, this definitely is where I'm thinking that in order to move forward, you definitely have to think about quality planting material. Yeah, you may come up with superior seeds, but seeds will be seed. It will still give you a less homogeneous performance than clonal issued plantation. So it's interesting because Dr. Ma was first talking a lot about agroforestry, and I put it in here because I think it's important, especially for smallholders. I think one of the problem with smallholders, well, before I get into that, let me talk a little bit about in Indonesia, Central Java. Teak is a very important species for smallholder in agroforestry setting. And as you can see on this slide, 55.9% of planted species is teak in Central Java. And timber apparently is a very, very important crop for the smallholders. So I think this on a global basis, the smallholders farmers teak planting comprise about 19% in Asia, 31% in Central America, 34% in South America. So we are talking about a lot of people planting teak as a livelihood. What is important though, we have to remember is I think I got this, which I thought was very important. This was a comment that was noted during the Laos Teak Forum, Sustainable Teak Value Chains for Sustainable Local Development, taken place in February 2020 in Vientiane. And one thing that they noted that smallholder farmer, if they receive technical know-how, high quality planting materials at lower cost, and a modest financial incentive, and so on and so forth, I'm going to read out, then I think smallholder teak plant production is going to be quite substantive in the production of teak timber down the road. So as a result of that, we have been also promoting to our buyers, how about trying out, giving some of this material to the farmers to determine how our clonal teak will do when, mixed in planting with other species. On this picture, there was a farmer who planted teak with arrowroot. I don't know if you're familiar with arrowroot. We eat it also. It's kind of a starch for thickening, sauces and so forth. And this is in Indonesia, and you can see the arrowroot is planted under the shade of teak trees. At the same time, in French Guiana, our collaborator being France, Sirac, they have this collaboration there. And we also asked the farmer to plant, with coffee and with banana. And here in Java, Southern Java, one very steep slope in an agroforestry setting, you can see the clonal teak doing very well, despite the slopes, the trees are still doing very well. Of course, they have to do things like weeding in the first year, but here don't expect the farmer to do any kind of pruning. They won't do that. But again, the acorns show very characteristic, very outstanding characteristics such as straightness, reduced lateral branching, high leaf density, and these all give a very impressive growth rate. And this, I think, photos were taken by Olivier Montuy during his visit there. Here in Ivory Coast, we also work with a group there, intercropping rubbers with teak to maximize the land use and return. This is in Ivory Coast. Here in Ecuador, the clonal teak under an agro-pastoral setting, and it works really well. You know, you can actually put teak clones at the border. We had the same thing in the Philippines. I sent out clones to the Philippines in a cattle farm. Teak trees doing very well. Here in Sabah, for sure, we have been encouraging oil palm plantation to do what we call border planting by using our clonal plots, and the teak trees grow very well. You know, if you are looking at the 25 years rotation of oil palm, similarly, you are going to get another cash crop from planting teak. So it works out really well. You know, it's all very good. And from here, we know a smallholder can definitely benefit from using clonal planting teak, clonal teak planting materials. Moving on now from smallholder, what would be the prospect for using clonal teak for large plantation company? As I mentioned earlier, in Australia, when we started out, we dispatched a lot of teak between the years 2002 to 2009. More than 1 million plants were dispatched to North Queensland. Now, all these areas were under ex-sugarcane. And at that point, during that period of time, sugarcane, the price of sugar was not so high. And that was the time when they decided that they should turn to forestry. And then they approached us and we sent them our clonal plants. So this is the initial setup over there. The company that bought from us did a spacing of 2 by 4 meters, whereby it can allow mechanization, where putting fertilizer or weeding is concerned, you know, herbicide application. You can have a truck just moving through among the rows. And thinning was done 2 to 3 years, you know, creating a spacing of 4 by 4 meters, and pruning was done up to the 5th year. Now, the area here is under very high precipitation, 3,000 to 4,000 millimeters per year within a 4-month period. And then another one, a lower precipitation towards the end of the year. Unfortunately, I think the company that bought from us invested. Despite having planted all these plants, I think they went under voluntary administration. So I don't really know what happened to the teak plot over there now. I'm sure they are still, our clones still, the trees are still there, except that the plantation, I'm not so sure. So it's a pity. Here in Brazil, we also work with Foresteca. Foresteca at that time was with the head lab. And then after that, another company came into the picture, Proteca. And these pictures are all from Proteca. I'd like to acknowledge them, especially Fernando Torres, for sending me all these beautiful pictures showing how mechanization of clonal teak planting is taking place in Brazil. And Brazil is very nice in respect to the elevation of the land. It's very flat. They are planting clonal 50-50, meaning to say they are planting 50% of our clones with 50% of new clones. Now, there's a reason for that. They don't want to put all the eggs in the same basket. So while trying out the new clones, they put our clone in place because they already know the performance, okay, of our clones. The climatic condition here is about 1,200 millimeters of rain from October to March. Temperature more than 22 degrees Celsius, and it can get up to 30 degrees Celsius. The soil you can see here is more of a clear, loamy soil with pH of about 6 and above. Here they apply what they call best management practices. They start shoot pruning from the age of 2 years, and they remove the shoot up to 3 meters, and then thereafter up to 5 to 7 meters. Pruning operation is never more than 50% of the height of the trees, okay? And a lot of epicomic shoots are removed in order to prevent, loss of energy by the tree in producing all this unwanted shoot. Now there are 2 thinnings done for under this management. I think they actually came up overall in 4 thinning operation, carried out every 2 years. And the spacing that they have used or they are using is 3.8 times 3.8 meter, as well as 4 times 4 meter. So the final densities after 2 or rather 4 thinnings is about 156 or 173 trees per hectare. So it's quite intensive, but they, I guess they know what to expect based on, the earlier work that they have done. So in, I think now, to date, they have planted more than 2,000 clonal teak plants in Mato Grosso. I believe the company will continue to plant. I think they have very good investor coming in. And I think based on the performance of the plantation, as you can see here, it's looking very well, very nice. So I'm sure the future of clonal teak in Brazil is quite bright. Moving on, in Cambodia, we had the opportunity to also work with a couple of companies there. Originally, the company that came to us was establishing plantation using seedling. Now the seeds were, I think, sourced from neighboring countries, and condition of the soil was not so good. Rainfall was very low, and so as you can see, the conditions are not that great, and there was much failure using seedling. So when they approached us to get our clone, again we did some kind of demonstrational plot just to be sure, and this was eventually there was a shift to clonal plants from our company in 2012. Here, this company is actually Teak Farm. I think Dr. Ma mentioned the same company earlier. They are involved in what we call precise propagation. You can see the sand or the soil is very sparse, very sandy. And if they are looking at a very fast rotation, I think it was mentioned they are looking at about, up to 3 years. But I think it would all depend. It could go up to 6 to 7 years. Here, propagation, precise propagation is applied to every plant. This company is quite good with irrigation system. This is 18 months after planting. The clonal trees look good, and we are happy to see that. Moving on, yeah. This is an overview of the plantation in Cambodia. DBH of 12 centimeter, height of 7 meters at 1.5 years. So the clonal teak trees are looking very good. So from all the slides of my presentation, we know that clonal teak can be a very high value and very important species for cultivation. I'm not going to go to all this, like they're easy to propagate and so on and so forth. What I'd like to bring to our attention as to why we think that clonal teak is going to be increasing in importance is from a biological perspective. Using vegetative propagation of teak, it doesn't show any C effects or within clone variation. Unlike some species that we had worked with, acacia, manju, we do not see any pleiotropic or abnormal phenotypic development, regardless of the age of the donor trees. It can be clonally propagated by shoot apical meristem, as I showed earlier. And this meristem, are quite vigorous in growth. And, I think we are quite confident that now with the technology that we have developed, we are able to introduce any teak trees regardless of their age for in vitro culture. So this is our conclusion from our, our early work on teak. Moving on, I think I've come to the last section. I know I've gone on quite a while. Sorry about that, but in fact, it's a lot to talk about. Covering 25 years of work, more than 25 years of work. So finally, last part of my talk will be on conservation of the genetic resources. We have always been very focused on the conservation effort to protect our genetic resources. We shall continue to gather and enrich our genetic resources, and this will be wisely managed as breeding population, and it will remain our priority. So based on the collaborative work with CIRAD, we have established 2 progeny provenance plots in Sabah. These are our correction. These 2 plots were established in 1997. In one side, it is on a hill, a slope, which is about 150 meters above sea level. pH 4.8 to 5.6 and sandstone soil, not very fertile soil. And this plot has 42 seedlots and comprising of 15 bulbs and 27 open pollinated families. Within, these trees were, of course, established in Ivory Coast within seed orchard. Now, don't worry about the information here. You can, you're welcome to get the information from it. Through me. We have already published all this information. So just quickly to let you all know that these 2 progeny provenance plots are where we have our collection of different different genotypes. So we have been doing thinnings, as mentioned. After 2 thinnings, one plot has 204 trees and the others about 240 trees. But of course, over the years, we are looking at about 1997. We are looking at about 14 years. Is it 1997? Yeah, 14 years. Rather 24 years almost. Of the age here, and we have done thinning. So the clones, the trees that remain, are now going to be our seed producer. We collect seeds from the 2 plots. And we have also been selling them. People have approached us to buy this from us. And at the same time, we have also been introducing these clones for in vitro culture based on the phenotypic characteristic. Here you can see these are all the clones that we have under tissue culture propagation. I think currently we have about 50 to 70 clones under production, and each of these clones, the background we have specified them is very, very important. That there's no mixing of the clones, that the background of each of these genotype is preserved, is documented, so that we know exactly where each of them go in the future. And this is, of course, to prevent risk of inbreeding. Again, there's a list of these clones in one of our publications. So we know from our work here how important it is for to have this clonal seed orchard, or in other words, we like to come up with the term convertible clonal test. Now, CSO issue seeds can be used for advanced generation of breeding to produce seed such as quality seed production, or it can be used as a clonal gene bank. And from the clonal gene bank, of course, you either get your wood-based population. Of course, you will always have pros and cons from such clonal test. So it's important that you plan out you do a proper layout of the clones. It is important that this CCT have high number of clones, and each should not be represented by 5 individuals to avoid inbreeding. So if you are talking about collecting shoot, you don't want the same trees to collect seed from the same trees over and over, and you want to promote crosses between this individual. So CCT are therefore evolutionary gene bank through the new seed issue genotype produced annually. So for higher reliability, the clonal test must be set up in similar environmental conditions as where the industrial clonal plantation would take place. However, we know from experience that setting such tests in different locations is not practical due to cost and time. So as a result, companies that have approached us instead choose to find environments that have similar requirements as those where our clones were found to be adaptable to. So this is quite crucial. For us, the Solomon clones have been observed to be well adapted to a broad and diverse range of environmental conditions. And this, I think, we have been lucky, so to speak, to be able to, work with such excellent clones under such a broad and diverse environment. So besides improving quality production and clonal tests, are therefore a judicious option for maximizing the return on investment from all these clonal test establishment. Jointly with CIRAD, we have continued to dispatch clone to a few places that are affiliated to CIRAD, to Gabon, Ivory Coast, French Guiana. I think the most recent one is 67 clones were dispatched to French Guiana early this year to set up clonal tests. So for us, it's useful and important that we are able to share these clones and to set up all these clonal tests, and just to find out, what the performance of these clones are, and eventually, if the results are good, these plots can be converted to CCT. So after several decades of expectation, we are pleased to say the clonal forestry has become a reality and is rapidly expanding. And I think it's here to stay. So I hope to see that on a global basis, if you talk about sustainability, we'll think more about clonal teak. But of course, you will also have to bear in mind that keeping the genetic resources rich and diverse is equally important. So with that, thank you for your attention. It's been a pleasure. It's a long presentation, and I'm sorry for the long presentation. But as I said, it has been a lot of more than 25 years of work here. So I think I worked quite a bit on trying to summarize as much as possible. This final slide here shows a couple of pygmy elephants in Sabah. They have come to look for food among the teak plantation. So thank you very much. Thank you to all the organizing committee for allowing me to present. Once again, thank you very much.
Thank you, Dr. Goh. We have one question from a couple of participants. In case somebody is interested in procuring your clones, how do they approach you?
In fact, I have been having quite a number of inquiries from India. And I always ask them one question. We need to get an import permit. So I don't know about the authority. Every time I ask for an import permit in order to do that, then I never get any response from the inquirer. So yeah, we can we are open to this. For me as a researcher, it's pointless to just keep your clone to yourself. I think it's important to be able to share. But on the other hand, it has to be a mutual understanding between both parties in the sharing of such genetics. So yeah, we are open to it. Definitely, it's not a problem.
Thank you, Dr. Goh. We will inform them accordingly, and then we will try to help them in getting this import permit so that they can get the plant.
Yes, definitely.
So thank you, Dr. Goh, for taking us through this journey. Though you say it's a long presentation, for us it was more of a story where
Yes.
you started off with the selection of the mother plants to standardization of the protocol, and then routing of the plants, and then their field performance. So I don't think anybody felt the tiredness going through the journey, that story which you presented here. It was an excellent presentation, and the information that you revealed, that teak does not require a distinct dry season. That has been an eye opener for us in India, because we always feel that we have to have a distinct dry season. So thank you for that information. I'm sure people who are growing teak will now be ready to grow it in any place and will not look for this dry and wet season. And the significance of use of advanced technologies, especially for the researchers, for proving the worthiness of the wood produced through tissue culture. That has also been well emphasized in your talk. So I'm sure the researchers who are going in for tissue culture teak will now take it ahead with confidence, so that they can use this technology for faster growth and then early harvest of the wood.
Yes.
Another highlight of your talk, especially in this webinar, is we have many participants from commercial laboratories. Your talk encourages all those people to take up tissue culture on a large scale, so that teak production globally can also be enhanced. Thank you so much, ma'am, for your talk. It was very enlightening. We enjoyed your talk, ma'am.
Thank you very much. Pleasure to share. Thank you.